Okay, hello and welcome to today's webinar. Today we're going to be talking about Conda. Before I get into that, I'm just going to cover, as I usually do with these webinars, just who I am and the types of activities that I'm involved with. I've been involved with bioinformatics for around 20 years, mostly in the genome informatics space. I have a background in pharmacology as an undergraduate, although that's getting further and further in the past. My main sort of interests lie within sort of like the HPC, Linux sysadmin, and the genome informatics pipeline building and analysis side of things. I'm also involved with training quite a bit. My background in terms of programming languages have sort of ebbed and flowed a little bit, and my main go-to languages at the moment are Bash, Perl, and R, but I've also dabbled with JavaScript, Fortran, and a few other things as well. The types of work that I've been involved with is in the area of genome assembly, gene annotations, and variant analysis. My PhD was in phylogenetics and molecular sequence analysis. I've done a fair amount of transcriptome assembly and transcriptomics, differential gene expression analysis, gene co-expression networks and things. I've also been involved with CRISPR studies and looking at genome diversity in general. So I recently left the University of Adelaide, and I've set up a business in genome informatics, providing consulting and training opportunities. The types of activities that I'm engaged with is around sort of freelance work in the area of pipeline development, optimization, and actual analyses, providing consulting or advice on experimental design, analyses, and various interpretations of those sorts of things, and training, whether that be one-to-one or small group or online or in person. These links at the bottom are different channels that you can contact me through. And so onto the webinar for today. So this is a bite-sized webinar. This is supposed to be a regular, generally a weekly webinar that's short, so 30 minutes long. It tends to be focused on a very small topic, so that we can cover it in sufficient detail in just the 30 minutes. And it's generally free for non-industry people. And you can access tickets for the different events at the link at the bottom there. That's just my reminder to switch the layout. So the key learning outcomes for today's webinar is that basically by the end of this, you should have some idea why you actually want to use Conda for installing software. Hopefully you already have an idea since you're here. You should be able to use Conda to install various different software packages, and I'll go through some examples. And to give you an idea of how you then go about using Conda to do what's called activate the environment, and that means to make it available to you to use the software that you've installed, and also how to actually go about finding whether the software package that you're interested in is actually available and what versions are available, etc. So we start with the problem of software installation, and if any of you have tried this in the last few years, generally there's probably three different viewpoints on this problem. So from a user perspective, generally speaking, if you can't get something to install in 10 minutes, you've probably wasted enough time, and if it still hasn't worked, you probably will give up and you probably won't ever come back to using that tool again. And typically people don't want to become experts in being a systems administrator, have to know how to compile stuff. It just becomes a bit of a nightmare very quickly. From a system administrator perspective, that's someone who looks after the computer that you might have access to. So that might be a high performance computing system, or it might be yourself looking after your own laptop. Generally speaking, you want to try and keep tools up to date, and this takes time and effort. And generally speaking, you want to be able to provide a tool say, a version of a tool like 1.0.3 of something available, you might want to make it available to the users today. You also want to have it accessible next month and next year for reproducibility purposes, so that an analysis can be rerun with the same versions of the tools. And you don't want to have users wait lots of hours or even days for yourself to install something, because you'll hold them up. And from a tool author perspective, you really want to have users spend time exploring the tool that you've developed, not to spend hours trying to install it and banging their heads against a brick wall. So all of these for different people results in lots of hair pulling and thinking life's not worth living and there must be a better solution. And in recent years, the solution or a solution is this tool called Conda. Okay, so that wraps up this first key learning outcome of why you might want to use it. It saves you a lot of hair pulling. So what is Conda? So Conda is a package manager. It's effectively an executable, a binary file that you can execute or run on a command line, and it essentially helps you to install, update, and remove software. But there are also these other things that you may or may not have come across, or you will come across if you've not used Conda previously, and that's this thing called Miniconda and Anaconda. And quite often it's a little bit confusing to start with. What are they? What's the difference? So Miniconda is effectively a minimal installation of Conda and some of the associated software and tools around that that help you to manage packages. It doesn't contain any software, any tools that you might want to use for analyses. Whereas Anaconda is what's called a distribution. It comes with Conda and all of the package managing things, but it also comes pre-installed with a bunch of software tools and packages straight out of the bag. And once you've installed it, you can access those tools and run them. So the analogy that I'll use sort of throughout these slides is this concept of a hardware store and tools, or a tool belt and things like that. Now you'll see that as I go along. So I've sort of stolen this idea from this website here, and there's a link here. It's a really useful website. It's got some interactive workflows that you can go through to sort of decide what you need to do, how you do different steps. So I'm sort of using this as a bit of inspiration, I guess, for the slides that I'm going to present today. So the very first thing actually that you want to do is, does the system you're working on already have Miniconda or Anaconda installed? If you're working on a high performance computing system, chances are it already is installed and you just want to use that. If you're working on your own laptop and you haven't installed it yourself, it probably isn't installed. So on a HPC system, one way that you could see whether or not it's available is to use this module available command and ask whether this module called Conda is available. This may throw back a list of different versions of Conda or Anaconda or Miniconda, depending on what's installed. And on the system here, I'm showing that there's a few different versions of Anaconda already installed. And so on this particular system, I can choose to load one of these Anaconda versions, which is this version 3/5.0.1. And once I do that, then Conda is available to me on the command line, and we can sort of proceed from there. So what if you don't have Miniconda or Anaconda installed, such as on your own laptop? You have two choices to install Miniconda or Anaconda. Anaconda is the one that has all of the software packages pre-shipped with it. There's about 150 packages. So one of the main considerations to whether or not you install Miniconda versus Anaconda is if you've got enough disk space. Now, 3 GB isn't that much, so typically most people will have that to install Anaconda. And once you install it, you'll have access to these 150 odd software packages straight out of the bag. Miniconda is the option if you have less than 3 GB. If you've got less than 3 GB, you probably aren't able to proceed with many NFCs. So the advantages with Miniconda is because it doesn't contain all of this software bloat of packages you may or may not use. It's quite lean and clean install, but you then have to go about installing the software packages that you need and want yourself. Now, that's not a bad thing because you're going to have to do that anyway. So my preference is that you go with the Miniconda installation route if you can, and always start off with a really lean, clean installation and then manage the packages that you need as you go along. So that's what I'm going to cover now, is how you go about specifying packages and installing packages. So there's a few different concepts to just cover before we get into actually installing Conda software packages. And that is this idea of Conda environments. And so coming back to this hardware example, an environment is a little bit like a tool belt. Okay, you create a new environment. It's empty. It doesn't have any tools in there, and you go about adding tools to that environment or that tool belt until you've got all of the tools you need to work with. So this can be done in several different ways. You can add tools one by one, asking for them to be installed. You can use like a little recipe type thing, which I'll come on to shortly, to describe what tools you want to install. Now, this analogy with a tool belt is quite nice because you can have different tool belts, each with different sets of tools. You might have one that's for cleaning, in this case, a little kid's tool belt. You could have all of these different tool belts, and depending on what job you want to do, you can just go get the tool belt or the environment that you want with those tools already available, ready to go. So how do you go about actually setting up one of these environments? So the way that I would recommend this is that you create this file. In this diagram, it's a list. So this is a description of what tools you're going to want in your tool belt, what tools you want available in your Conda environment. And you use this list to define that and to set that up. So the actual Conda command that you would use to do this then is this Conda env create. So this is creating an environment. Prefix here tells you where you want to store it. And so this is like the name of the tool belt, the envs directory here. And we have a file, and we use this file to define what tools we want to install. So let's take a look at that file. Sorry, just before we get to that, where you actually store these environments is really can be really important. My recommendation is to store them as under an environment directory in whatever project working directory you're using. So in this case, I'm just setting up a project working directory with this env subdirectory. And to create an environment file, I'm going to call it rnaseek.yaml. So this is just a YAML file. It's a specific file format. And I'm going to show you what that looks like now. So the very first line here should be the name of the environment. So this is the name that you're going to call the environment when you want to use it. So in this case, rnaseek. Then there's this block here called channels with a list of different channel names here. So I always use these three. There's very little reason to go to other channels, and these channels is where Conda will look for tools when it's looking to install them. So think of this in the hardware store type of thing. It's different hardware stores that you might go to find a tool. And then there's this dependencies block here, which lists the tools and the versions that you want to install into the environment. So this is the tools, and this is where Conda is going to look for those tools when it's installing them into the environment. Okay, so that's how you go about using Conda to install software packages. And so once you've set up that environment, you then have to go about using it. So this is a bit like taking that tool belt that's got the tools already installed, putting it on ready to go. Okay, the command that you need for that is this Conda activate and then the path to the environment. So in this case, I'd installed it under this envs subdirectory. And laser pointer on this envs subdirectory, and the name of the environment was rnaseek. And so by doing this, we can activate this environment and get ready to using the tools. So we're ready to go. We want to use the tools. Let's see what's available. We can do commands like star in this context because we've installed star as a tool. And so when we run star, we can see that it's ready and available to use. Okay, so the next thing that you really might want to think about is, okay, I now know how to use this stuff. Where do I go to look for software packages that are available for use in Conda? Is my favorite tool available, or version even? Oh, my apologies. The next step, sorry, before we get into that is the idea that once you've finished with the tools, you want to get rid of them. You want to take off that tool belt, put it down. You don't want to be weighted down by all of that environment that's available. And the command that we use for that is this conda deactivate. Now, to update environments, so for example, if I just head back to this YAML file, in here we had these versions, and SAMtools here is set to 1.9, and we might want to know, okay, what happens then at some future point in time when that version increases? So there are new versions of SAMtools available now that's beyond 1.9. And you can update your environments at any point, which is akin to taking the list that you used to, this YAML file that you used to create the environment in the first place. You find that, okay, there's a new version of Hammer or Star or SAMtools, and you update it in this environment file here, and you get a new tool, new version of that tool within your environment. The command that you use for this is conda env update, and again, we provide it with a prefix to say where that conda environment will reside. We point to the YAML environment file where we've changed the version of the tool, and we use this prune. That means that if the new version of the tool no longer has a dependency on a tool that it previously had a dependency on, it will remove those out of the environment, making sure that it remains clean and as lean as possible. So what tools are available? There's two different paths that you can take. My favorite, doesn't require remembering any syntax on the command line, is just to simply go to anaconda.org. And you will get to this website here, Anaconda Cloud. It's a very lean interface, and there's basically a search bar at the top up here. And so you can enter into that search bar anything that you want. So in this case, I was looking for a tool called Genome Tools, searched for that, and you get provided with a list. So you have three aspects to this list in terms of the packages. You have this first part here, which is before a slash, which is what's called the channel. Now, if you use that YAML file layout that I showed previously, it already includes this Bioconda channel, which is where the tool is going to look when it's installing something. And then you have the name of the tool here. So in this case, there's a genome tools - genome tools, and then there's a version 1.6.1. Some useful information in here is that by default, this list is sorted by the number of downloads. So generally speaking, you don't have to look very far beyond the top 1 or 2 lines to identify the tool that's most relevant to you, unless you're trying to use something very obscure. So here we can see there's nearly 10,000 people that have downloaded this Genome Tools package. So that's likely to be the one and the most recent version as well. You can access a search page like this just with a tool with a URL like the one shown here. And the other way that you can do it is via the command line using Conda. So you can do Conda search, and unless you use wildcards like these stars here, it will search for the exact phrase that you looked for. So if I hadn't got these stars, or at least the star on this end here, it would only return this first one here because it would be searching exactly for genome tools. So we add in the wildcards here to make sure that we capture something that's a bit more fuzzy to what we are actually looking for. And again, it returns a list similar to what's seen on the website with the versions, the channel that it's available in, and so forth. Okay, so that now means that you should have some idea about how you go about searching for your software that you might want to see if it's available for Conda to download and install. There's a huge, all of the popular software, bioinformatics software is available for install. There's very few that are not, although the latest versions of some things might take some time to come through and be available in Conda. So that's pretty much the conclusion of today's webinar. So the idea is that next week there's a PacBio continuous long read webinar where I'll talk a little bit about that technology, what it is, what it can be used for, and how it differs to other technologies. And again, just to sort of sell my business for paid work, there you can engage with me on any of these things, whether it's actual doing freelance work, actually doing hands-on analysis, pipeline building and things like that, or if you want to tap me for advice, consulting, then feel free to get in touch. Or if you're interested in holding some sort of training event, I'm looking to develop some events over the coming weeks to hopefully do in-person training. And those will typically be up to 20 people per session. And again, here you can contact me by this email address or this LinkedIn page. And so that's it for today. I will take questions because we have 5 or so minutes left of the session. So if you want to use the chat feature, I will hang around and wait for any messages or questions that you might have. I think I have Terry typing a message. So feel free to head to the chat and type a question if you have one. So Terry's asking about Python 2 versus Python 3 packages. Sounds like you've probably been burnt at some point. I'm not a Python expert. My experiences with Python are possibly like yours, in that there are some tools that will depend on Python 2, some on Python 3. Now, once you start to install multiple tools into an environment, you can run the risk of getting conflicts, and this definitely happens around Python version dependencies. So as tools have sort of moved on and are supporting Python 3, they might drop support for Python 2, or if you've got a legacy tool that only supports Python 2 and not Python 3, and you try and install those in the same environment, it can get messy quickly. So my main approach to try to circumvent those issues is really to try and keep the environments as lean as possible. So I typically try not to install more than half a dozen tools into an environment. Now, you can have a different version of Python installed within the environment itself. So in that YAML file, you can define a Python version within that. Otherwise, it inherits the same Python version that you are running Conda in say in the sort of in your normal operating system environment. Okay, so I have a couple more people typing, so I'll wait for those. Okay, so one question here is when using Conda, do you need local copies of files or configuration files and things like that that come with the tool, if that tool requires them? So typically those will be available. You might have to, so those will typically be available and installed as part of the package, assuming that the person that developed the Conda package knows what they're doing. Now, you may have to provide the path to that file explicitly. So you might need to do something like a which command to find out where the binary file is for that tool and use that as a way to try and figure out the location of where all the files are that was installed as part of that software. So if like that GTF file is part under that subdirectory of that tool, you'll be able to sort of find your way through there. Okay, and another question is about Conda versus containers, Singularity, Docker, etc. Okay, so there's Conda and containers serve two sort of separate things. One, Conda is more about managing the software environment of the tools. Containers more about managing the operating system environment in which those tools operate. So if you think you can use Conda on Windows, Mac, Linux. You can manage that software that you want to use, but it doesn't. It's sort of independent. It's dependent on the operating system on which you're running. Containers allow you to abstract out that container outside that operating system, such that you are managing both the operating system environment and the software environment that you're using separately. And you've got two different tools that you're using to manage that. One's the containers and one's Conda. So they're similar concepts, but addressing two different parts of, I guess, reproducibility in terms of constraining your operating system environment and constraining your software environment. Okay, we have about 1 minute left, so I will hang around, and if there's any more questions, feel free to type or get in touch following the webinar. Okay, I don't see anyone typing, so we will wrap this up and I will end the meeting. Thank you for attending and stay tuned for the next one, which I hoping will be next week, but it may well get bumped to the following week. Okay, thanks guys.